Okay. Hi, guys. How are you? First off, thanks to Miss Richardson for the invitation. I am really excited to come and share a little bit about my path and what I do with you all. I do hope that you get at least one thing out of here to hold on to, and you can take some good notes. So I'm going to share my screen. Mm. So, Miss Richardson, okay, hold on one second. Do you see my PowerPoint?
Yes.
Okay, the PowerPoint and not the speaker view, correct?
Exactly. We can see you also. So we can see you and we can see the PowerPoint. You just may
Oh,
not be able to see us.
okay, great. Okay. So, um, let me start off by saying that I don't follow directions very well, and that's a mark of a good scientist, just so we're clear. So I was given a task to kind of come to you all to talk to you about biotechnology and about how I use it in my day-to-day life, how my career has really been about biotechnology, and so I'm going to do that, but I'm going to do it my way. So I am Brandy Huderson. I am an assistant professor of biology at the University of the District of Columbia. And so let's get started. So, a little bit more about who I am. I am unfortunately not a native of the DMV. I'm actually from New Orleans. New Orleans will always be home for me. I am also one of two girls. So, I think you all met my sister, the other Dr. Huderson, last month. And so you already know that we're both scientists. We sit in a lot of the same spaces. We do a lot of the same work. Our degrees are different, but in the end, we've been together a lot of the places in our journey. We are the only two scientists in our family, and um, we've been doing this for a while. And doesn't matter what happens. Whenever we go home, the least little thing that happens, people are kind of looking at us like, "Is that right? Is that the truth? What is this new science thing we're talking about?" And it's been especially the case in the last year since we've been dealing with the pandemic. So I have always been a science girl. I have always had a natural curiosity about me. I've loved taking things apart to see how they worked. Um, it just always made sense to me, and it was just really easy. The science and the math, it clicked and made sense. Other things didn't quite make so much sense to me. History, art, but science and math always stuck out. They were just things that I could do very well. So, just to quickly tell you about some hobbies that I have. So, even in my extracurricular work, even in the things that I do for fun, they still all seem to have a STEM component to them. So, for example, I love to sew, I love to knit and crochet. These are things that on the surface don't really seem like it, but they do have a lot of STEM in them. They have a lot of math and they have a lot of engineering. They're about repeating patterns, they are about being able to create something from nothing. You're rearranging materials to make them into something else. And that really is at the heart of things. It's a lot of coding, it's math. And then I love to cook. And I'm showing you one of my favorite things that I made, which were Snicker bars. This is nothing but chemistry, right? This is figuring out how much of salt, how much of baking powder to put together to get the desired effect. So this is really kind of where my life sits. It sits at this place of STEM. So, a little bit about my education and training. So, as I said, I'm from New Orleans. I received my bachelor's of science from Xavier University. I have a bachelor's of my degree is biology pre med, and we'll get to that in a little bit. After I completed my bachelor's degree, I went to the University of New Orleans where I received my master's of science and biological sciences. And let me say, I have this highlighted because this is really where my biotech career started, my understanding of the capacity um, of what we could do with science. And I began to learn techniques and skills that kind of set me up for the rest of my educational and professional career. After I got um, my master's degree, I went to Virginia Tech, which is in Blacksburg, Virginia, only about 4 hours from here. And I actually entered to my graduate program in a program called VT Prep Scholars. It's a post-bac program. We can talk a little bit about that. But basically what it is for those people who didn't graduate with a 4.0, but who still had potential, um who still showed an interest and a capacity to do research and to do work. And so they give you a year to kind of catch up and prove yourself. And that's what I did before I entered into my actual PhD program. My PhD is in animal science with a concentration in dairy science. So I got to work with cows. Those are my babies. And while there, I added on to my agricultural work with the dairy science with molecular cell biology and biotechnology certification. So I had an extra piece that allowed me to basically tell the world that I am competent in this separate skill set. So once I finished my PhD, I then did 2 postdocs. So getting a PhD is similar to a medical degree in that once you have your degree, you have to do a training period after that. And so for medical school, it's called a residency, but for a PhD, it's called a postdoc. So I did 2 postdocs. I went back home to Tulane University School of Medicine, did a postdoc, and it was in cancer biology. I focused on steroid receptor activation. And then I did that for 2 years, and I came up here to the DMV and went to Georgetown for 2 years and still focused on cancer biology. When I finished up that last postdoc, I began my academic career at the University of the District of Columbia, where I am a tenure track assistant professor of biology. And I know some of these things I've said don't quite make sense, and I'll explain them as we go through the presentation. So just to kind of give you an understanding of what I do my day-to-day. So as I said, I'm an assistant professor and I'm tenure track. And so what that means is that I am, my position is one that has potential for promotion. And so what I do is I spent the first 5 years of being a professor kind of gathering my data, doing my research, teaching my classes, and performing service to my institution. And what I do is I put this together in a packet and I submit it to my administrators and say, listen, this is all the work that I've done. This is how I've contributed to this institution. This is what I'm good at. This is what my peers have said that I'm qualified to do. And they evaluate it. If they deem it good, if they think that I've done a good job, that my service is good, my research is good, and my teaching is good, then I can be promoted. So I'm starting here as an assistant professor. So if they approve my tenure package, then I get to become a tenured associate professor. So it's like a promotion. And at this point, I have a permanent job. So then the way that it works then is you, as an associate professor, you continue your research, you continue your teaching and service, and you can be promoted again to professor. So I have a PhD, and PhDs are research degrees. They are not professional degrees. So I am a doctor, but I'm not a medical doctor. That is a professional degree. I am not a dentist. That is DDS is a dentist, it's a professional degree. So these are degrees where you go take the professional degrees, you take classes, you do internships, you do work in the field, and then you go out and you become a practitioner, meaning you're seeing patients, you are actually working in the field. Research, what we do instead for my PhD was I was in a laboratory, I was conducting experiments, collecting data, and at the end I wrote a dissertation or basically a very big research paper. I then submitted that research paper to a committee that read over it and said, good, maybe not. So once they give their final approval, you then defend your dissertation in front of a crowd of people. It's a public event. And what you do is you explain to the group what you've done, what you found out. You give them some background about your information, and then they ask you questions. And you have to be able to answer these questions to show that you actually know what you're talking about, what you've done. Once all of this is done, they give you a big red check, good job, and you receive your PhD. At UDC, I have a small research lab where I focus on steroid receptor biology, and we use molecular techniques to do this research. So I say I have a small lab because it's just me and my students. So there are other labs that may have the principal investigator, which is the person in charge of the lab. So I'm the PI of my lab. They may have a lab tech or a couple of lab techs. They may have postdocs and graduate students. In my lab, it's just me and my students. My students are undergraduates for the most part. So they are students who are attending UDC. So they spend about a year with me working on a research project. So when they come in, they've taken classes, so they have the basic knowledge, the theory knowledge. And so while they're in lab with me, they get to actually learn how to do these skills with their hands. So thinking about some of the things that you all do. So I think I'll talk to Ms. Richardson, and you all are familiar with things like PCR and Western blotting and RNA. So they actually spend time in the lab with me doing the research, collecting the data, and in the end, they do presentations. What everybody wants to know is how much money you make. in this field. So I am in academia. There are different parts of, there are different not different places for biotechnology. So I sit in an academic space, which means I work in an academic institution. I am on a 9-month contract. So what that means is that for 9 months out of the year, I am obligated to be a part of my institution and to function as a full member of that institution. The other 3 months are mine, and I'm free to do with them as I please. So for my 9-month contract, I make about $70,000. During my 9-month contract is when I do my teaching, it's when I do my research with my students, and it's when I do my service. The other 3 months are technically supposed to be mine to go off and do as I please. I can go work with somebody else in another lab. I can continue to work on my own research, or I can take the summers off. As an assistant professor, you really are on the grind to collect as much data as possible so that you can publish your papers, and so that when you get ready for that tenure package, you are prepared with all of this scientific research that you've conducted. So most of my summers were spent in my lab doing research. I have students that come into the lab. There are summer programs that bring students, whether they are undergraduate or high school students, through the DC SYEP program, Summer Youth Employment Program. So they can come into the labs, they do some research, they get hands-on experience. At the end, there's a poster presentation. And if I actually have enough data collected, then I can actually publish a paper. Um, a lot of the time that I spend as an assistant professor is spent writing and working on science-related grants. These are grants that I use to support the research that I do. They're used to support the students in my lab. They're used to support us attending conferences and publishing papers. I also work with other people on their grants as well. So these grants can range from something as small as $50,000 to upwards of $5 million over the course of 5 years. And I say small with $50,000 because if you think about the work that you do in a laboratory, it's really expensive. So a small vial, for example, of antibody with maybe 200 mils in it can run you $250. And if you need 12 of them over the course of 6 months, I'll let you do the math. So it adds up very quickly. So $50,000 is a relatively small amount of money. To get 5 million is kind of like what you're aiming for if you're in a research lab. So just a little bit to talk about my actual research program that I have. So like I said, I have a steroid receptor biology program, and steroid receptors are the actual receptors for steroids in your body. And I'm talking primarily about two steroid hormones, estrogen and androgen. And androgens, most people know them as testosterone. So I'm interested in understanding how these two hormones work through their receptors in the mammary gland. Specifically, I'm interested in how they work in normal breast tissue, how they work in diseased tissue such as cancerous tissues, and also how they work in tissues that have been primed by testosterone but are now receiving estrogen treatments. So think of the breasts of trans women. So my lab uses molecular biology and molecular genetics based techniques. So we do a lot of cell culture work, and our cells are transformed. And this is where the biotechnology really starts to come in. So we're using transformed mammalian cells. And what that means is that we've gotten cells that have been taken out of a mammal. It could be a mouse, a human. We have some hamster cells as well, and they've been transformed. And what I mean by that is that we've used a virus to introduce genes into these cells that allow them to replicate in a plate. We cannot just take the cells out of your body, for example, put them in a dish and get them to just keep growing on and on and on. We have to actually treat them or transform them so that they are going to be viable over a long period of time in a dish. It is possible to take cells out of the human body, put them into a dish and grow them, but they tend to die out very quickly. So you have to be very quick about what you want to do with them. These transformed cells, I can get a cell line, I can get 5 cells. I can let them grow, reproduce in a dish, and I can have this cell line for the next 20 years as long as I take care of it. So we focus on transformed cells. Some of the techniques that we use in my lab are things like Western blotting, real-time PCR, transient transfection. And so transient transfection is when we take a cell and we introduce a gene or a genome into it, and we allow that cell to express those genes, but they're only going to express them for a short period of time. After that, the cell is going to be like, you know what, this doesn't belong here, and it's going to kick it out. So that's a transient transfection as opposed to stable transfections, where you can actually add the gene or genes into the cell's DNA, and they continually express it. They're going to divide, and the next cells are going to express it as well. So we've done a lot of work with transient transfections. Immunohistochemistry. So this is using antibodies to actually locate certain proteins in a cell. We do site-directed mutagenesis. So we can take the code for a gene and we can change it. We can change it however we want. We can make it into something different, and then we put it back into the cell so that the cell can then express this mutated gene. This is one of the ways that we use technology to figure out what genes do. So by turning them off, we can look into the cell and see, okay, so what's changed when we turn this gene off? Or we can turn on a gene and we can say, okay, so what has changed in the cell when we turn this gene on? We also use light and fluorescent microscopy. So all of these things sound great, woo-woo, but what do they actually look like? So this is how my science looks coming from my lab and the research that I do. So these are some pictures of the work that have come out of my lab, either out of my lab now or out of my research as out of my graduate research. So these two figures are actually published figures. And so one of the cool things to look at is here we're looking at a cell line that we actually created. So we've used not immunohistochemistry, but immunocytochemistry. So what we're doing is we are probing cells instead of tissues with antibodies looking for the expression of particular genes. So in this case, we have here two cell lines. These are cancer cell lines. They're breast cancer cell lines. They're very common. So MDA, MB231, MCF7s, very common breast cancer cell line. So what we did is we took the MCF7 cell line and we actually removed a gene from the cell. We removed the estrogen receptor gene from this cell. We turned it off. And what we wanted to see is that if we added back in a mutated estrogen receptor gene, what happens to the cells? So here's our, where'd my mouse go? So here's our original cell line. These are the original MCF7s. This is where we knock the gene out. So we turn the gene off in the cell. And here are the two cell lines that we created by replacing a mutated estrogen receptor. So we mutated the receptor in two places, and then we put them back into the cell line. So what this picture does is it shows you actually a bunch of techniques all in one. So site-directed mutagenesis, we are doing immunocytochemistry, we are doing fluorescent microscopy, we're doing cell staining. So we're using all of these different techniques in this one panel. So we looked at the cells to see how they look different based on what genes they were expressing. These are different antibodies that we use to probe the cells so we could look and see how they looked. So this is just an example of how my lab is using different biotechnology techniques. So here what we have on the top right is this is the Western blot. So here we were looking for expression of a certain protein. These two actually go together. So these are the cells, the same cell lines. So here we were looking at the entire cell, taking a picture of it. Here we took all the proteins out of the cell. We treated them with an antibody, and then we expressed them on a piece of film. So we don't need to know all the details that went into it. It's a published paper. If you're burning to read this paper, I can definitely send you the link, but the cool parts are right here on the screen. These are the pictures. So these are some other things that we've done in our lab. So this is what we call a colony formation assay. And so when we grow cells in a plate, especially cancer cells, sometimes they tend to grow in clumps. And so these clumps represent cells that have been reproducing and reproducing and reproducing. So what we wanted to see is if we treated our cells with any type of chemical that inhibits cancerous growth, what happens to their ability to grow in these colonies. So what you see at the bottom left here, these 3 purple wells, we treated the cells with an inhibitory compound, and we wanted to see the effect on the growth. So you can see that in the first well, all the way over to the left, there's purple dots in there, and those represent colonies. But as you move from left to right, you'll see that there are more colonies. That's because the first well, they responded to the treatment, and so there's less cell growth, less colonies formed here. And then as you move over, you'll see that those other wells didn't respond as well to the treatment, and so they continue to grow. I'm keeping my eye on the time. I get really excited about this, and I won't get to the rest of the presentation. So here is a mouse that I actually was using in my research. And so what I did is I actually used those cell lines that I created in these top panels, and I inserted them into a mouse to see how well these cell lines could create tumors. And so this kind of fuzzy, but I drew circles around these tumors. These are pretty big tumors. These are huge tumors. This was very exciting for me, not so much for the mouse, but to see that the cell line that I actually created actually works in a living organism. It actually does create tumors. And so now what we can do at this point is we can go in and we can start treating these tumors in an actual living cell system, an organism, and see how this organism responds to treatment. Because how cells respond in a dish and how cells respond in an actual body are going to be very different. So this is one of my preliminary experiments. I created the cell line. I got it to grow. I tested it. I injected it into the mouse. I got tumors, and now we're just ready to go. So this last picture over at the bottom right is actually a picture of a calf mammary gland. This is from work from my PhD. And we were looking for a particular enzyme. So again, we were going to use immunohistochemistry. IHC is what we call it in the lab. And so all of that brown that you see in that picture, that's the actual enzyme that I was looking for. And this was pretty cool because this was the first time anybody had actually shown that this enzyme was present and active in a tissue. So very excited. Something else to look at in this picture is you see these big circles, those big white circles in the picture. That's actually because those are lipid cells. And so that big drop in the center is actually a drop of fat. And so the drop of fat is so big that it actually pushes all of the components of the cell to the side. So all we really see in this picture is the cell membrane, and then this big drop of lipid. And you can kind of you can compare that to some of these other pictures in the top left corner where you can actually see like a really nice nucleus in the center of the cell. You don't see that in these lipid cells. Okay. So all that's great. It sounds wonderful. And I encourage everybody to go to a lab and do research and create mice and create cell lines. But I want to bring us back a little bit to reality because I don't want to give you the impression that it's all been hunky-dory and great and I am just this super smart person that soared and did all these great things. It's not true. I mean, I'm smart, but I didn't just soar. So in the very beginning, when I was like 5, I thought I was going to be a veterinarian. I love animals. I love mammals. That's what I wanted to do. In fact, at 5, I decided that I was going to be the first Black woman veterinarian. So just to clue you in, I am older than you all, but I'm not old enough to be the first Black veterinarian. There were plenty of Black women veterinarians before me, but I was 5. So what happened, though, is a lot of the people in my family, my community were like, we don't really do dogs. Black people don't have animals. We don't fool around with that. That's not what you want to do. You want to be a doctor. So I was like, okay, fine. Mind you, I was 5, but I was like, okay, fine, whatever. I want to be a doctor. So I thought I wanted to be a medical doctor. I went all the way through high school telling everybody I was going to be a medical doctor. I went to Xavier University of Louisiana, which is #1 in the country for placing African Americans into medical school. And I was going to be a pediatrician, and then I learned about something called neonatology. These are the doctors that take care of the sick neonates, sick brand new babies. I was going to be the first Black woman neonatologist. Again, that was not even an option at that point. They had plenty of them before me. But right around my 3rd year of college, I began to have second thoughts. One, hospitals smell funny. And I was like, I don't like this smell. And another thing that I began to realize after doing some volunteer work and shadowing some doctors is that babies come with parents. Parents are adults. I was like, I I don't think I really like adults. So I began to try to figure out what it is that I did like and what I wanted to do. So some side notes to this side note. I played around in high school. That and they tricked me. I was in the gifted class, so my classes were weighted. So if I earned a B in a class, it wasn't just a 3.0, it was a 3.33. And I was like, okay, cool. I don't have to worry. My GPA is high enough. That's great. When it got time for me to go to high school, I mean to college though, they unweighted all of my grades. And so I actually went from having like a 3.1 or something or a 3.0 to actually having like a 2.5. And I was like, oh my God, what happened? But I goofed around, right? I wasn't serious. I wasn't paying attention. And that's not for you to do. Um, you should not do that. But it's just to give you a realistic view of who I am. I am a person. I made mistakes. I didn't pay as much attention as I should have at all points. But that doesn't mean that you won't be successful in the future. I got to college. I kind of played around in college too, except for two classes, organic chemistry and ecology, because I got mad at organic chemistry because I got a D. And I was like, whoa, that's not okay. So I took it again. I got an A. And ecology was a course that I took my senior year of college. I was again goofing around, and my teacher pulled me to the side. And he was like, "Just so you know, if you don't pass this class, you're not graduating." I'm sorry, sir. My dad has already planned for my graduation. If I don't graduate, I'm gonna have a problem. So we had to get serious about ecology. I graduated with 2.0 GPA. When I actually saw my GPA, the first time I paid attention to it was when I graduated, and I was like, "Oh my God, what am I gonna do?" Um, for my master's program, I didn't actually apply. I took the GRE, I got decent scores. They were like, hey, if you come in, if you apply, we'll take you. That is how my career began. There's some people, my sister, for example, who was very, she was very focused and directed and knew what she wanted to do and was on that path. Um, I was not. I was just kind of like, I don't know what I want to do, but I'm going to do it. I'm going to get to it. So don't panic if that's where you sit. Not yet, anyway. So one of the things, though, about all that playing around in all of my experiences in high school and college, and even in graduate school, got me to realize that um, one of the things that I didn't know was my options. I didn't know what I could do. I didn't know what I could do with my degrees. And so it got me to thinking as I'm moving through my career, as I am getting serious, as I entered into my PhD program and I began to fully understand what I was doing, as I entered into my position as an assistant professor, I really began to stop and think about what it is that I wanted to do. How did I want to do it? And along the way, I remember having conversations with people who really had no idea about science or what it could do or what it meant. They were very confused. They had heard the wrong information from another person who'd had the wrong information from another person. And I knew that I had a unique skill set and that I understood all this biotechnology. I understood how science was being manipulated and used to improve things and make things better. And so it really got me to thinking about how I wanted to use my science. So beyond just being an assistant professor, I've really spent a lot of time putting my biotechnology knowledge and use into STEM education programming. So I work on creating programs, after school programs, Saturday school programs, that not only focus on children, but also focus on adults and providing them with experience, hands-on opportunities to learn and use some of these technologies. So for example, doing things like extracting DNA, things like running a Western blot or doing real-time PCR to help them understand a little bit better about the science. I'm okay with you not liking science. I'm not okay with you not understanding science. I'm not okay with you being afraid of science because you don't know. So what I want to do with my biotechnology knowledge, with my biological knowledge, is I want to be able to give people enough information for them to be informed and to understand what's going on around them. And I think this last year with the virus, the pandemic has really shown why we need to have a working understanding of biology, of chemistry, of how we use these things, the biotechnology that's being used, so that people aren't afraid. So this work also made me wonder about who decides what we learn, who decides how we learn. And so I took time off from my assistant professorship to do a fellowship with AAAS. and I'm placed at the National Science Foundation. The National Science Foundation is a big federal grant. It's a small agency, but it does a lot of federal grants, and they focus a lot on STEM education. So they focus a lot on educating not only people in schools, but also the general public. And so I was really, really interested in that. I also use my biotechnology understand knowledge and experience as a consultant. So what consultants do is they go into a setting or an organization or a business, and we help you figure out how to create programs, how to run a business, how to do things better. And so I consult with teachers. So for example, let's say Miss Richards wanted to figure out how to put um, how to do transfections in her classroom. So I would consult with her. I would come in and bring my knowledge that I have about this technique. So I would help her design the programming. I would help her design a timeline to make sure she get the work done. Help her come up with the curriculum to teach to the students so that they could actually understand and get something out of it. I also use my biotechnology expertise in the space of diversity, equity, and inclusion. And this is kind of a hot topic right now. And so what I'm doing in this space is I am helping people to see how things like systemic racism have impacted communities of color, particularly Black and Hispanic communities, how this technology has been used against them, or how they have not been allowed to access this technology. So I help people to see this and to redesign programs that are surrounding biotechnology to make sure that everybody has access to these programs. And I also participate in grant writing. So I'm going to skip ahead a little bit because I'm looking at the time. So I use my expertise as with biotechnology to help people design grants. So one of the things you have to do in a grant is you have to explain the techniques and experiments that you want to do. Some people don't really know how to do that very well. So I can help them with that. So now, what about you all? How do you all take what you all are learning right now in this biotech high school, in this biotech curriculum, and how do you move forward? So first things first is to acknowledge that you all are in a very unique position. Most of my students that I see in my freshman biology class haven't even taken a science course in their high school careers, right? That's the first thing I ask. Have you taken biology or chemistry? And I get a lot of students that are like, no, I didn't take those courses. So you all are already ahead of the curve. You're getting this really crucial hands-on experience with biotechnology, and so you're prepared. So the important thing to remember, though, is that this is the beginning of your journey, right? When you finish high school, this is step one. This is not the last step. And you do need to continue your education. Now, how you choose to continue your education is completely up to you. Whether you decide to do vocational training or career and technical training, or whether you decide to go to college and graduate school, it doesn't matter. But you do need to understand that taking your educational career to the next step will help you. Being able to expand your understanding and knowledge of what you're doing, it's only going to help you as you move forward in your career. One of the things that you do have to think about as well is you have to think about how do you want to use your knowledge. So I use my knowledge in very different spaces. So I work in an academic institution, but I also do consulting on the side. I do STEM programming. So I'm in a very much so educational space. But there are other places that you can use biotechnology expertise. So you can do the academic or nonprofit research route. Or you can do for-profit research. So if you wanted to go work for Johnson and Johnson, Moderna, Pfizer, these organizations, that's another option for you as well. Let me make a note here. Just because the company is nonprofit does not mean that you do not get paid. Nonprofit is just about how they're organized. They still pay. You'll still get paid for your expertise and your knowledge. You can use your biotechnology experience and expertise in policy making. So figuring out how we set up science policy. What do we prioritize? What do we need to know? What does the government want to spend their money on? That is a great need right now. Consulting is always an option, as well as government work. So the federal government runs national labs all around the country. They hire people that have different levels of expertise and experience. And so that is a very viable option for you in terms of a career. So with that said, you don't have to be the best. I think I've shown you that I was not a 4.0 student. I did not graduate top of my class. I'm not proud of that, but it is just a fact. but you do need to be prepared. And so you don't even have to know what you want to do right now, but what you do need to be thinking about are things like, what is your passion? What are you passionate about? And what are you passionate about not doing? So even if you don't know what you do like, you should know what you don't like. And you should be keeping track of that as you think about what it is that you want to use all of this biotechnology expertise at this point, and how you want to apply it to your careers. The most important thing that you can do, regardless of whether you know what you want to do or not, is you can take advantage of opportunities. So opportunities that come up, such as summer programs and internships. These are ways that you get to figure out a little bit better about who you are as a scientist, as a biotechnologist. You figure out what it is that you like and you don't like, and it's also a way to get exposed to different fields and different job possibilities. So you'll have a better idea of how you can use your skills. You all are in high school, but think about taking courses, for example, at UDC's community college. I have to say UDC because, well, I work there, but at a community college. So take an introductory biology class, take a biotech class. These are types of things that can help you figure out what you're interested in. It also is college credit, so it may be able to be transferred to the whatever school you're going to attend for college. If you're not sure if you want to do college or if you want to do more of a training program, this may help you figure that out. So these are opportunities for you to do that. As you're thinking about summer jobs, as you're thinking about if you're going to work while you're in high school or college, think about working someplace that will allow you to use the skill set that you have. So instead of going to McDonald's, and there's nothing wrong with that. That's where I worked for many years when I was in school. But think about possibly getting a job with in a research lab, so an academic research lab, or think about in this area, there are lots of pharmaceutical companies and biotech companies that have internships. So think about applying to some of those as opposed to just taking a random job. These little experiences add up to a big pot of skills. And so these are things that you can then take with you when you're looking for your jobs. Learn more about biotechnology and the people that are doing biotechnology. LinkedIn is a resource. I don't know that it's appropriate or a good idea for you guys in high school. I don't know how the school set this up. If you all should have your own individual LinkedIn pages, but maybe if Ms. Richardson, for example, wants to create a LinkedIn page for the school, and they can recruit people to come in they can connect to people who are doing biotechnology. And this is another way for you to figure out who's doing what and what the possibilities are. You can also use Twitter for this. Think about hashtags, hashtag biotechnology, hashtag Black in biotechnology, hashtag woman biotechnology, hashtag short in biotechnology. It doesn't matter. But Twitter is a really great resource. Also think about mentors. If you see someone who's doing something that's really interesting and you want to know more, contact them. Send them an email, tell them who you are, and just ask them if they have some time to chat with you, or send them some questions via email that you would like to get answered. I think that's a really good way for you to find out how people are using what people are doing. And I would also suggest group informational interviews. So 2 or 3 of you all get together. If there's somebody you're interested in finding out more information from, get 2 or 3 of your classmates together, set up a Zoom meeting, and you all just kind of having a sharing meeting. Nothing formal. We just want to know a little bit more about who you are, a little bit more about what you do, if you have any advice for us. And I think a lot of people in this space would be more than willing to actually provide you with any type of information or guidance. Okay. So thank you. I appreciate it. That's one of my cows. That's my babies from my PhD. She's bad, but that was my sweetheart. So, all right. Do you guys have any questions? Are you guys still there? I don't know. How do I stop sharing?
Yes, we are here.
Okay. I'm going to stop sharing my screen. Okay. What is the biggest challenge I've faced in my career? Thank you, Ms. Gamble, for giving me the hardest question starting off. I think that there are many places where I felt unsure about myself. I felt insecure about my abilities and what I knew and what I didn't know. And that persists to today. I th- uh, that has probably been the biggest challenge that I face, is being secure in um, being secure in what I know and who I am and what I have to offer. I have spent a lot of time in school. I have a lot of knowledge, but I'm not always comfortable in that. And so that is something that I work on a lot. And the way that I work on that is with peers, it's with friends. And it's kind of reminding myself that, listen, you've done a lot of work. You've been a lot of places. And although I don't know everything about everything, I do know a lot. I know a little bit about a lot of different things. And so being comfortable in my knowledge has probably been my personal biggest challenge. Um, yeah, that's been my biggest challenge. So another question is, okay, so would you say you want to make this a long term? Whoa, it's moving to like I'm 60. So, I do want to be an educator, and I'm very interested in providing and being an educator. I enjoy doing STEM education, and I really can see myself doing this for a very long time. I think the spaces that I would do this in would change. Do I plan to be a professor for the rest of my life? Probably not. There's so many different places for me to do STEM education, and it's so many different levels. I would love to see more of, I would love to experience more of those different places. So, possibly working at the National Science Foundation so that I can do STEM education there, thinking about running a STEM program where all I do is just train students all the time, things like that. Those are, that's kind of where I see myself. Um, but I also love research. I also love the bench. I would love to like set up a little research lab in my backyard where I could just go and tinker around and play with cells all day long. So I'm looking for opportunities and things that will allow me to balance those two things out. But I don't necessarily see myself remaining in academia for my entire career. There's just so many other things to do. There's so much other stuff to get in to get involved in. Do I have a soft spot for animals? It is ridiculous. All I do every day, not all day, but all I do is watch videos about chimps and elephants on freaking Facebook and Instagram. Hour of my time gone. Actually, what I would like to do is take probably about 6 months off and go to there's a chimpanzee and orangutan reserve conservation site in Africa where you can volunteer, and that is on my list of things to do somewhere, to just spend 6 months playing around with some orangutans and some chimps, and maybe do a elephant sanctuary while I'm there. I am not interested in treating anything or anybody clinically, though. I don't want to provide medical care for anybody. I don't want to do that. Um, so, having a career in the same field as my sister, have you ever had to ask her for help or advice? And does that make your relationship closer? First off, I'm the oldest. She comes to me for advice. That's not true. I have. And it's actually, it has made our relationship closer. Um, and she's a great resource to have because she thinks about things very differently from me. And so, um, it's nice to have her there to kind of balance me out. It's You really do need to have some type of village or support system in place. And this is regardless of what your career will be or where you're going to go. You really need to have people surround you who can provide you with the type of support that you need at different times. So my sister is able, she's here, she's a sister, she provides sister support. We work in the same spaces, we work together a lot, and so we can provide professional support. Um, it's really been nice to have her around. Um, Definitely. And we give each other advice. So she comes to me to ask me about things. I come to her to ask her about things. But it's really been a big help, I think, in both of our careers. Um, so I put 6 years retirement age. Okay, so I don't know that I'll actually retire. I'm a busybody. I enjoy doing things. I enjoy working. I enjoy what I do. So 60 is really close for me. So I don't know, but we'll see. I don't know. what are the different types of work you do in small versus big animals. So my big animal work would be with my babies, my cows, and so I look primarily at their mammary glands, and uh, the physiology of the mammary glands. So how they're maturing, how they're working at different age stages, um, puberty versus not puberty. Um, if we gave them this type of supplement, what happens to their mammary gland development? Small animal work is a little bit different because actually in small animals, I'm not really interested in how their bodies are working. I'm interested in how their bodies are working when I introduce something different. I do not do a whole lot of animal work anymore. It got to be too much putting down just animal after animal after animal for me. So um, cells don't bother me, but I killed a lot of cows in my PhD, and it really didn't make me feel good. I've done my fair share of mouse work as well, and I was just like, this is enough. So I'm taking kind of a pause from that. There's some projects that I want to do that require mouse work, and so I'm working myself up to get back to that. But for right now, I've just had to take a pause. Um, do I enjoy Animal Planet? So I got rid of cable, so I don't actually have access to Animal Planet, which is why I supplement with Facebook videos and Instagram. Um, and I've worked with several types of animals. I've worked with large primates. I've worked with horses. I've worked with cows. I've worked with sheep, chickens, pigs. They're complicated. And mice. Um, Cows are by far my favorite. Cows are very large. I've done some work with horses. Yeah, horses. I said horse. Cows are by far my favorite. Those are the ones that I spent the most time with. But all animals have personalities. The difference is when you have a 2,000-pound cow or heifer who has a personality. Heifers being, that's an actual scientific term. Heifers are young cows who have not actually given birth yet. Um, and they decide that they are upset and they want to show you that they're upset, or being covered in poo, which is what happens when you work with animals. Lots of poop. But cows are by far my favorite. Um, yeah, the girls are sweet. They can be sweet, they can be moody. They're kind of easy to work with. Yeah. So, yes, when you work with animals in research, there are going to probably be some times when you are going to have to um, you're you're going to have to euthanize them for the purposes of the research. Uh, I know it sounds terrible, and it is terrible, but that's kind of where we are right now. We're working a lot on creating models so that we don't have to involve so many animals in our research. But at the end of the day, models only go so far, and we still have to go back into the animal. Oh, yay, Courtney! I love the girls. I do. For a long time, I did not eat beef. Um, I do now, unfortunately, but before I didn't. It was just too much. It was a little bit too connected. Do you like the classroom more than working with animal research? And what was your favorite research? Um, I enjoy the classroom, but I treat my classroom like an extension of my research lab. So we're there so that I can give you information, so that you can have an experience. I don't treat my classroom as if I'm here to just throw information out at you. So it's really an extension of my research lab. Um, the most exciting research that I've done, honestly, has probably been with the cows. I was raised in New Orleans. I lived in the city. I had never seen a cow up close before, other than like dinner. And so when I got to the farm at Virginia Tech and I saw all these cows, and I learned so much about agriculture, and I began to actually understand about my food and understand about animals, it really changed the way I looked at the world. And so to this day, that's probably been some of the coolest research that I've done. I do not drink milk. I have never really drank milk, though. It's just not a favorite drink of mine. I think a lot of the information that we have about our food is not necessarily based in truth. And so I would encourage you to do lots of reading and research on that. That is a whole nother presentation about understanding food and what we've been told and what's actually real. So I don't necessarily think there's anything wrong with milk. unless you're lactose intolerant. You just shouldn't have milk if you're lactose intolerant. And that's pretty much what I'll say. I don't like the taste of milk, so I don't drink it. I thought I saw someone's hand up, but my screen is moving, so I can't find them anymore. I love dogs. I had two dogs. I had two dogs for a very long time. They were mutts. Got one from a shelter. Got one off the streets of Atlanta. They hung out with me for 14 years, both of them. So future doctors, what kind of doctor? Do you want to be a PhD doctor, a research doctor? Do you want to be a medical doctor? Do you want to be a dentist, a pharmacist? I think it depends. But what I will tell you is that an investment in your education will pay off. It pays off not in a guaranteed job, not in a guaranteed salary. It pays off in guaranteed opportunities. You say you want to do surgery. Oh, Lord, child, you're going to be in school forever. But again, I think an investment in your education, whatever that education may look like for you, pays off. I think it's worth at this stage in your career, I really think it's worth looking into what being a medical doctor means. And so talking to medical doctors, if you want to do surgery, talking to a surgeon. I really recommend shadowing a doctor, just so you can see what their day-to-day life is like. It is absolutely not like what you see on television, right? Grey's Anatomy is not realistic. So figuring out what it actually looks like. And also figuring out what other options are for surgeons. So suppose you go to medical school, you graduate, you do surgery for 5 to 10 years, and you're just like, I'm ready for something different. So thinking about what are your other options. Always keep in mind what your options are, because you never know what will happen. You may wake up one day and be like, I don't want to see another person on a table again. What can I do with my degree? And there are tons of things that you can do with a medical degree that don't involve seeing patients. So what are they? Are those things that also interest you? So I definitely recommend for anybody to not only think about or not only focus on what it is that you do want to do, but think about what are your other options surrounding what you want to do. I think that's the last question I saw. Ms. Richards, are there any other questions? Ms. Richardson, I'm sorry. Are there any other questions that you have?
Oh no, this was fantastic. I think Shayla had her hand up. I'm not sure if she's still on.
Somebody had. I saw somebody's hand raised. I'm so sorry.
Yeah, I had my hand up, but I had put my question in the chat.
Okay. Did I answer it?
Yeah, you did.
You don't sound confident in my response. I can change my response or make it better if you need
I'm
me
trying
to.
to scroll back up to show you what I said, but I was just basically asking like, what's the difference between like working with small animals versus big
Oh,
animals?
yeah, the difference. So the difference is what you do with them, but it's also how you handle them. Um, Big animals, you need to be very careful. I love cows to death, but they can squash you. They can harm you. A small animal can cause minimal damage. Um, I prefer to work with large animals, but I like bigger animals in general. Um, and it's really the type of research that we do on different types of animals. Smaller animals, we tend to use them kind of as a vessel for the research. We give them things, we put things in them, and we look at what happens. In larger animals, we tend to do more observational work, right? So it's not so much manipulations of their bodies that we're doing, and so it's a little different in what we're in the way that we treat them. Large animals require different types of care. They require more space, so you can't put a cow in a research lab, right? So you need a farm. Whereas mice, rats, hamsters, you can put them into smaller spaces. Um, you have to be trained to work with any type of animal. So you'll have to do some type of animal training, animal care work, just so that you know how to handle them, how to treat them the correct way, how to identify if they're ill, how to how to be able to assess if they're happy or they're sad. And I know that sounds crazy, but it matters, right? These are research animals, but we still want to treat them with dignity and respect. And so there's a lot of effort that's put into making sure that they get, for example, with mice, we put enrichment toys in their boxes. So they get wheels, they get balls. With cows, we don't necessarily do enrichment, but we make sure that they have ample space to move around. We make sure that they have ample space to work with, to kind of socialize with each other. Sometimes, depending on what's going on, they may get a toy or two, but cows really just want to eat all day, so we don't really have to worry about that. So it's just, we do treat them differently. We use them in different ways, and they have different, they do have different needs. Okay. I don't know if you guys have another class at 1 o'clock, so I don't want to run over, but if there are more questions, I have no problems answering.
Yeah, it is about 1 p.m. So scholars are probably going to head out to lunch. Go ahead and thank Dr. Huderson for her time and her presentation. Thank you very much.
I hope this was helpful and useful.
Absolutely. And do you have, Dr. Huderson, contact information if students have questions or if they'd like to?
I do. I tell you what, I have a LinkedIn page, and I don't know what it is. So if you go to LinkedIn and put my name in, though, you can find me.
That sounds great. That's perfect.
So yeah, my Gmail account is perfectly fine. If I don't respond immediately, if I don't respond within 24 hours, email me again. I might have missed it. So that's the only thing.
All right, that sounds great. And scholars, if you have questions at all for Dr. Huderson, you can reach out to me. Awesome. She just dropped her email in the chat, so you can reach out to her directly if you'd like to do that. And thank you for being here today.
Thank you so much for the invitation. I really appreciate it. Thank you.
That was awesome. Thanks again.
Thank you.
Yeah, I just wanted to reiterate, thank you so much. I really enjoyed that personally, and I'm sure the kids did as well. But even just some of the stuff you did, I was like, oh, next year when I teach about lipids, of course I should show them fats. Like I need to show them that picture of the fat in the cell. So just thank you.
I'm excited. I was trying to figure out how to work in some pictures. I was like, all this talk doesn't make any sense if you can't see it. So I'm glad.
No, that slide was great. Thank you.
Good. Thank you.
Definitely. So yes, Dr. Huderson, we will let you get on with your day. I will definitely be in touch and send you a copy of this recording so you can check it out. And we appreciate your time.
Thank you so much. You guys have a great day and enjoy your weekend.
You as well. Take care.
Bye-bye.
You too.
Thanks.
Have a good day too.